Today we're going to talk about automated chemical storage and high throughput compound synthesis using the Hamilton Verso and the Microlab Vantage. So to get started, this is the actual system that sits in my lab. On the left is the Hamilton Verso, and on the right is the Microlab Vantage. We use the Microlab Vantage for chemical synthesis. Vantage is, of course, is Hamilton's liquid handler, and on the left is their storage unit, which has historically been used for compound management, but as you'll see in this talk, that we use it for different purposes. So our Verso is an M3 Verso. It stands for 3 modules. Every module holds roughly 2,000 trays. Each tray holds 5 SPS footprint racks. There's obviously they can make high density racks and whatnot like that, but ours uses particular SPS footprint plates. Ours comes with a 4-channel star picker, which these systems no longer come with those pickers because this was one of the first ones that they sold in the US. Ours comes with a standard IO, which is a manual input and output where you can walk up and do deposits or walk up and do requests. Ours has a robotic IO, which Hamilton calls a handoff. That's another spot that connects to our Vantage. Ours has a particular capacity of 95,000 vials, and ours is stored at room temperature with no temperature controls, no humidity controls or anything like that. We have a Microlab Vantage. Ours comes with 8 nanopulse channels for individual like cherry picking and compound movement. It comes with a 96 core head for bulk dispensing. It has a track gripper for moving racks and plates. It has dual entry exits. We use the entry exits for storing consumables and for microplates for our runs. And this also has Brooks Fluid X decapper for our 6 mil vials that you'll see that I'm going to talk about. Has a Hamilton shaker. Has a J-Chem heat block for heating compounds or heating reactions when they require it. And it has a working volume of 1 to 5,000 microliters, although that can be customized for everyone else's needs. Our particular needs are in that volume range. And ours also comes with a heat sealer. So that's my spiel on their instruments. I get paid 1% commission on that. I'm just kidding, I really don't. I don't work for Hamilton. But the reason why I wanted to start off with this is because both of these instruments are very expensive. And to buy these equipments, there's a lot of justification that has to go into this. So if any of you are ever considering something like this, I'm sure you have gone through this process. But to get to why we bought this, I have to give you a little bit of history on Neurocrine as a company. In 1993, Neurocrine was founded. That's when the company got started. We are a company that focuses on small molecules and the neurological and endocrine-based disorders. Between 1993 and 2006, our research group had screened 78 targets. 5 had made it to phase 1, 6 had made it to phase 2, and there's more phase 2 than phase 1s because we did some in-licensing in that time, and 2 had made it to phase 3. 1 had made it all the way to the FDA for what we assumed would become a drug. In 2000, we prepared for commercial launch for a sedative hypnotic similar to Ambien, but between 2007 and 2010, the FDA decided that they didn't want our drug. They gave us 3 non-approvable letters, and we had downsized greater than 90%. We were over 700 employees, and we dropped down to 58 employees in that time. For our research group, we literally dropped down to 9 people. We had 3 chemists, 3 biologists, and 3 engineers. Luckily, though, Neurocrine did decide to keep us as a company or a research group so that we could rebuild, because whenever you get that small, right, there's the chance that they could just close down research and sell the company. But they saw that there was a path for us. And our motivation at that time, because we had gone from being successful, even though we didn't have a product, but we had lots of investors, they were excited about our potential. Our motivation was to be more efficient, and the only way to be more efficient was to increase our probability of success of coming out with a drug by optimizing the lead optimization cycle. So for those of you in research, you know that coming up with a preclinical compound is your ultimate goal. You get an indication and you need a chemical starting point that you can develop and create analogs around to eventually build in the potency, to dial out the side effects, and to build in stability and stuff like that. So on this particular talk, where I actually am where I focus on the concepts or the chemistry, so molecules and synthesis. And as you can see, the other parts of this process, like assays and data, those have been optimized. Those robots, the Vantages and Stars, were originally built for things like that. But what we decided to do was, what we needed to do was optimize that cycle time. So the key to developing drugs is that cycle time was too slow. We only screened 78 targets. We needed to up that number in order to come up with drugs. So how do we minimize the design and data generation? Or how do we minimize that lag time? And that ultimately, if we could minimize that, would lead to us being more productive. And just like high throughput screening and compound management, where they treat their process like a manufacturing process, chemistry decided we needed to do the same thing too. So we needed to identify the steps in design, synthesis, and testing that really add value, and find the processes that don't add value and eliminate those, or at least optimize them. So for us, the obvious low value work that we came across was reagent management. Every company has a manual, every company has a chemical storage area where it's all done manually. We had a group of 3 people dedicated to that, and obviously when we downsized, we went to none to manage that, so we had to do that. And we figured, we knew that that process was so labor intensive because all the bottles were different sizes and whatnot, that just checking inventory, just inventorying was a big time sink. At the time, at least in publications, what we read about for other groups to optimize that, we saw that some had outsourced their entire chemical management to like Aldridge. The problem with sending off all your chemicals to Aldridge is you have to wait for them to ship it back to you. So there's a big lag time that can be anywhere from a day to 5 days, especially with chemicals because of their hazardous nature. So we decided we needed to minimize the turnaround time by utilizing automated on-site storage. So what we did is we borrowed the concept from, at the time we had purchased a compound management system, which is the NatureCal MatriStore. Our system was designed to store final compounds. It had a capacity of 30 million compounds, so it was huge. It went up 30 feet high into the building. It was temperature controlled. We kept it at -20 because everyone was concerned about the freeze-thaw cycles with final compounds. It could store a wide array of SPS footprint plates, so tube plates, 384 well plates, fixed plates. It could do pretty much anything we used in high throughput screening or compound management. And the advantage of it were it could run overnight, although our particular MatriStore didn't really run overnight because of all the bugs. But in theory, it should have things like error-free picking, which we had changed because before that we were doing everything by hand, and as you know, doing things by hand, you can introduce errors. And it was designed to be the perfect system for long-term storage for compound management, so we figured, why not do this for chemistry? So the big leap that compound management groups made to become efficient was to store everything in solution. From DMSO, dry powders, going from dry powders to solutions made it much easier because then you could automate the liquid handling portion of it, where there's very few automation tools out there to deliver solids. So we figured, why not store our chemical reagents or reactants in solutions so that they could be liquid handler ready. Most reactions from small molecules are done in solution anyways. We typically weigh things out that are neat and then dilute them because you don't really want 2 solids. You can't really react 2 solids together efficiently because they're not the molecules aren't interacting until they're dissolved. So because we decided to do this, we needed to just find a universal solvent, just like compound management had done with DMSO. And then this process, if we could get it to work, would vastly reduce the time to synthesis. And in the big picture, it would vastly reduce our time in that iterative cycle of drug design. So obviously there are huge concerns with storing chemicals in solution. The first one is, are reagents stable in solution? It's the same question that compound management had for dry samples in solution, and it's probably a bigger question for reagents because reagents are designed to be reactive in nature. The next concern that we had was if the chemicals leached out because they're reactive, they could possibly erode the plastic storage, so the racks that they're in and the tubes that they're in could leak. And at the time, we only had plastic tubes that we could use to store these things in because no one made chemistry-specific tubes. And following along those same lines, if the chemicals could erode the plastic storage, then they could possibly erode the metal storage trays and the automation inside of the matrix store. The reagents could also, along those same lines, the reagents could also contaminate final compounds. So there were a lot of risks when we were testing this. Not only were we messing around with a multi-million-dollar storage unit, but we're also possibly messing around with our final compounds. But what the logic we went with is we weren't going to be building, we weren't going to be hiring chemical reagent managers. We weren't going to be hiring hundreds of chemists to do the same work that we were doing and when we failed. And the bigger question about universal solvents, or the chemist's natural concern was, a universal solvent probably won't work in all reactions that we do. So that was in 2005 or 2007 when we had gone through the layouts and we were rethinking the process. Fast forward to today, and here's what we've learned. That first concern, the vast majority of reagents are stable. We still use some of those reagents we plated out 10 years ago in reactions today. And we know that they work because we have mass specs. We know that they work in reactions to create final products. We have NMRs. Not all reagents are amenable to solution-based storage, and those reagents are typically the very reactive ones like isocyanates or sulfonyl chlorides, those types of things. And those types of things we do offline. We still do those by hand. We settled on N-methylpyrrolidine, or NMP, as a universal solvent. The obvious choice was DMSO, which we tried. DMSO did solvate most reactants, but the problem was when we needed to heat it, it decayed rather quickly and it made our reactions pretty messy. An alternative to NMP was DMF. That has similar properties to DMSO. That formulated amines, so it ruined our amines and that made them unusable. And then following DMF, we tried NMP, which surprisingly was a very stable universal solvent. And then we also found that NMP doesn't get in the way of a lot of the reactions that we do. So alkylation, Suzuki's, things like that, NMP works. We also learned since the reactants were still stable, they didn't contaminate our final compounds. And since they didn't contaminate our final compounds, they certainly didn't contaminate our robotics. We also learned at the time when we were aliquoting out reactants, we aliquoted them out to 96 well mini tubes. We learned that that was a very expensive process. And the only reason why we aliquoted is because that's the way compound management did it, because compound management used single-use tubes. But for reactants, we didn't necessarily need that. So we decided that we didn't want to spend $50 a plate for microtubes. And we learned over those 10 years when we were picking those reagents from our matrix store that it vastly improved our time to synthesis. So another thing that we did is after we standardized reactants in solution, we also standardized our bulk solvents. So in this picture you see here, this is actually our chemical storage area. And as you can see here, most of these bottles are sorted based on size. And they're also sorted in general locations. So if I wanted a barcode 1177, and it's in this drawer for section A1, I would have to pull out every single bottle just to find it. And if I was lucky, I might get on the first one, might get on the fifth one, but there are very many times when you have to look through this entire thing just to find one bottle. So it's very inefficient. And if any of you have ever ordered chemicals, you realize that you can order 1 gram of chemical from 10 different vendors, even the same vendor, and you're going to get a different bottle like every single time. So it's just not an optimal process. So at the same time, what we did is we standardized our bulk storage, our neat samples, into 20 ml scent vials. And the reason why we went with 20 ml scent vials at the time is Biotage had just come out with an evaporator that could evaporate solvents out of a 20 ml scent vial, and we figured that that was the best way for us to store bulk reagents if we ever needed to dissolve them, transfer them into 20 ml scent vials, because we could dry them down with that Biotage V10 evaporator. So this is a grid system, very similar to what you see in a microplate for compound management, where everything has a position and a location. So the way we would say, we would define this shelf as 1, we would define the shelf as 2. And every single one of these locations had like A1 or A2, depending on where it was stacked. And then obviously these trays have individual locations as 2. So even just going to standard containers without automation vastly improved our storage. So at that time, as Neurocrine rebuilt, after we had learned these things, we decided we wanted a chemistry-specific automation platform that could store our reagents, because it had already proven to be cost-effective to do this instead of hire reagent management people. And then we also wanted an automated synthesis platform as well. So, and we also wanted to move away from compound management that was set to specific vials and all that stuff. We wanted to build a system just for us. So we also decided if we were storing our chemicals that were reaction ready, why couldn't we just integrate it to a liquid handler? So it was one of those more efficiency steps where instead of having a separate storage unit that you have to go get something and then go place it onto your Star or Vantage, we just figured why not hook them up? It would make that process a little more efficient so you could work through the night. Another key factor is we had to drive the automation from our internal database. We couldn't afford to hire a whole bunch of automation experts, so what we needed to do was build a platform that everyone could use. And so along those same lines, we couldn't just have specific trained experts that you made a centralized group, because if they left or if they went on vacation, everyone would have to wait on that person. So if everyone used it, it would be much more efficient. So our requirements at the time for automated storage and synthesis was it had to be worklist driven, which is very important in this talk. It had to have bulk dispensing for co-additions to our reactions like bases and whatnot. And it had to have independent liquid handling capability, since we were doing cherry picking from individual reactants from our Verso and mixing them. It had to have a versatile deck so that we weren't interchanging out the deck. What I mean is we had to have a large deck, so we bought the 2-meter Vantage so that we could define certain positions for certain reagents all the time. And then we would also define a couple different spots as variable reagents. It had to be able to run overnight, because if you're buying automation, you're much more efficient if you can run 24 hours a day versus 8 hours a day. And again, it had to have the ability to move plates, so that's where we bought the track gripper, so it could grab plates from the Verso and move it to the Vantage. Had to be able to shake these reagents so that it can help get them into solution, since high concentration reactants can sometimes crash out of solution. It had to decap and recap, since we weren't using single-use tubes, we were using tubes that needed to be opened and closed. And it needed to seal, and it needed to heat, because there are quite a few reactions that need heat. So what we settled on was the Vantage and Verso. And this is actually my Vantage, and it looks really freaking pretty, because my lab is really messy, but Photoshop does wonders. So anyway, so again, this Verso stores 95,000 vials, which for us, I think we internally have around 60 to 70,000 chemicals. So we wanted room for growth, and it had to be modular, because the way we designed things a year ago or today are probably going to change in a year or two. And that's historically how it's been at Neurocrine. I'm guessing that's how it is at most companies. So with being modular, if we ever needed to make changes to it, instead of buying another one, we can just add to it. It had to be an open system, so the requirement was every chemist should be able to use it. And chemists using automation is like trying to feed a child vegetables. Like automation and chemists just don't go together, right? So that requirement might have been the toughest one. but that's where the work list came in. So work list, what I mean by that is we use an electronic notebook that's attached to our LIMS system. That LIMS system needed to be able to give instructions to the machine for it to run. That way, whatever variables we were typing into the system would be fed into the machine, and the machine could sort out where to put things. It needed to perform a wide variety of reactions for final step chemistry. We do about 8 different types of chemistry. We're not doing novel chemistry on this. We're doing proven chemistry on this. So it needed to do all of them. And basically, when we built the system, we learned that it could run for 96 hours straight with the EEs that we had inside that held the tips and the plates. And so that was actually huge. That was a huge bonus that we surpassed. So just to give an example of how we would make this system run and how we would be efficient is Neurocrine worked on 30775 here 10 to 15 years ago and developed, or 20 years ago, we were big in CRF, just like most companies. And this is a compound that we published. This compound is broken up into 3 parts, 3 main components. The top, the alkyl region, the core, this bicyclic region, and the bottom, which is this boronic acid region. Typically, the way we would have done this prior to our layouts is you would assign 1 chemist to 1 compound. As you can see, in this ideal situation, it takes 6 days to go from this starting material to this starting material to this intermediate template here, where on day 7, the final step chemistry, they would put on 1 amine top. So in ideal situations, this would take 7 days. More realistically, because yields and reaction success, this typically takes anywhere from 1 week up to 7 weeks. What we decided to do when we downsized and rethought things, we thought we had to do convergent synthesis. And what do I mean by that is we work as a team. Instead of assigning 1 chemist to 1 compound, we assign multiple chemists to multiple portions of the compound. So in this case, we would assign a few chemists, 3 chemists, to work on different cores here. They would all then put on the different amine tops, and then they would then pass off those bromo-alkyl templates off to me to do the final step. And in that final step, I would do all the different types of bottoms that we thought were necessary. So in the convergent optimization here, what we would have done is we would have said, how would we break this molecule down? Let's ask all the relevant questions at the same time. So what we would have done is scour our database or scour the publications to decide what was going to give us the most likely chance of success. So the first thing we do is we look at the core. That required the most chemistry. Once we figured out the core that we wanted, we would then put on the alkyl tops, and then they would hand off the 3rd step to me. And that would be how we would design the molecules in a nutshell. So on that bottom region, this is a Suzuki reaction where we take this boronic acid and attach it to the aryl bromide, and then we end up with a carbon-carbon bond. So how we would do this through our database is we would search core limbs. It's hard to see in the back, but this is a list of boronic acids, and this is exactly how it looks in our database. We would create a list and save that list in the database. We would then look at the cores, and here in this scenario, we have 12 different intermediates, and we save that as a list as well. What we then do is we give the machine all the instructions. So this is actually hard to see back there, but this is our database where we put in the variables of how we're going to do this reaction. So here are the different volumes of list 1 and list 2. Here is where we put in the list names. Here's the different trough additions that we would add. So one of these trough additions is aqueous potassium carbonate, and the other one is ethanol. And these positions are assigned to fixed positions on the deck. This would then enumerate the samples and give you final products. And when it gave you this final products, it would create a work list that's a flat file that's basically headers and all the information that we just input there, and it would send that to the machine. And since we wanted to minimize the complexity of this is where Hamilton's expertise came in, and this is where all that development time came in, is we built one assay. They call assays like program methods. Assay is a definition that they use. I like to say methods, one robotic method for all the compound synthesis we do. So if I were to do an alkylation or a peptide coupling or anything else, it would still be done through the one method on the Hamilton Vantage that I have set up. So all the instructions are in that work list, and those instructions could be heating, sealing, whatnot like that. And again, the Vantage deck, we have individual spots where we use labels to say this spot is NMP, this spot is Unix-based, this spot is triethylamine. All those spots match up to our electronic notebook. The heating and the shaking and the time and the sealing are all worklist variables, so not all reactions need heat. If we wanted heat for 30 minutes, we would just put in 80 degrees for 30 minutes. And all that is recorded into our notebook, which is something all scientists do anyways. Our database can mix 10 reagents per well. The Vantage can actually mix more, but our limitation here is the database. And then our tips, which are in those EEs, we have 50 microliter tips and 300 microliter tips. Those are auto-selected based on the volume that we're using. So that kind of intelligence was built into this assay too. We also have 1 mil tips on the deck. And we used CCD liquid detection to optimize the heights of pipetting, because since we're going to be cherry picking from tubes, you could have one tube with 1 mil and another tube with 5 mils and another tube with 4 mils. You can't set fixed heights. So that's where Hamilton's liquid level detection technology helped us out in that aspect. So going through the process, this is the manual chemistry process. So luckily we had a volunteer, Nick Pagano, who did this. And he's going to, so this is highly sped up, but this is how he would find his inventory. He would have to print out all the vials that he would want and then select which ones he wants and circle them, and then he needs to go locate them. So he's going to be famous for being Mr. Inefficient. So if you ever come to Neurocrine, you can make fun of him. But no, so these are all reaction-ready solutions before we built the Verso. And as you can see, he's arraying them into a 12-well trough. That way he can use a multi-channel to dispense them. But this is what we defined as low-value work, because when he's actually doing this hand pipetting, which is what a Hamilton Vantage could do, he could have been designing new compounds, right? Or he could have been coming up with new targets or anything other than hand pipetting. So this process took him about 2.5 hours, the total process of going through the inventory sheet and whatnot. And from here, he has to walk over to a different lab, to a heat sealer, just to seal a plate. And then he has to walk to a different lab, to a different heater, and that's where he finishes. So as you can see, that's a very inefficient process, and what you don't see is him putting away the reagents, because again, that's really low value work, and surprisingly, our head of chemistry does that for us. But that was because he knew we were going to evolve into the automated platform. So next, which is a professional video done by Hamilton, is the system that we built together. So as you'll see, it does picking. So in our case, we use 6 mil Fluid X vials for reaction-ready solvents, or reaction-ready solutions. The 1 dram vials are used for high weight final compounds, and then 20 mil scent vials are used for bulk chemicals. So this is just an example of how that would work. And then this is the robotic handoff. It goes straight to the Hamilton shaker, where it helps put those solutions, enables those solutions to be fully liquid. Then goes to the decapper, moves the rack back up to the deck, where it does the reagent pipetting. And again, all this is work list driven. And as you can see, it's not linear. It does what it thinks. It picks from the most optimal fashion in terms of where it thinks it should go. And then heat seals it, and then heats it. So it's that same process that Nick did, but to do one plate just like Nick did, this system does it in 30 minutes. And again, I don't have to do it. No one else has to do it. All they have to do is input this information into our database. That automatically sends the work list over to our machine. All we have to do is start our program and hit go. So the results of the Versa Vantage integration is we learned that if you want to standardize chemicals, you have to standardize the containers. It's exactly what the SBS Society did for the SBS plate. Once they created the SBS plate 20 years ago, they could then create automation around it. So if we can standardize chemicals in 20 ml scent vials, which can then go into an SBS plate, that's the way to go for automating chemical storage. So we learned that by automating chemical storage, it saves a ton of time. So if we wanted 100 different chemicals, that whole process of finding the chemicals, weighing them out for 100 different reactions, and then putting them away could be several days. So by eliminating those several days, that automatically shortens our iteration time of that lead cycle, of that lead identification cycle. So far, we have 35,000 reagents in our Versa, which seems small, but as we use chemicals from our old storage, we're weighing them out into 20 ml scent vials, and then also making solutions, reaction-ready solutions. We take that extra time and we focus on designing higher value reagents that we think will be active against new targets. And we right-size final compound synthesis. So chemists work in millimole scales, where biologists typically work in micromole scales. So when we were doing milligram scale, we were making thousands too much material for the biologist. So it was making a headache. It was a headache because we would make so much that we would have to store it in compound management. And that comes with its own set of problems. We miniaturize our chemistry to 384 since we now use the Hamilton Vantage, which has the modern liquid handling techniques. We could do reactions down to 2 microliters, which is very small. It's an open platform, so chemists at Neurocrine actually use this system. The system wasn't designed for me, it was designed for them. When we're actually doing synthesis, it doesn't stop us from using the Versa. The Versa can be used to still walk up and do manual picks. It doesn't interfere with the integration on the Vantage. We use this system to support internal live projects as well as building our own compound library. So instead of buying compounds to add to our library that we don't own, at least we don't own the IP to that structure, we build molecules ourselves that we can own. And then not only that, by having the molecules and the starting materials, if these things ever do show up on a screen, we then have the starting materials to make it again and get a dose response on a fresh sample, which is what pretty much every research group does. So the Versa Vantage combo really allowed for us to increase our efficiency. So as Neurocrine has grown, we're now over 40 in research, but over the past 2 years, we've added no chemists, and we still have no plans to add chemists. And while that might sound like this has replaced chemists, it's actually allowed us to hire more biologists, more purification people. Our purification group is going to triple this year just to keep up with this compound purification. And our biologists, we still can't hire them fast enough. So if any of you are looking for jobs, there's jobs on Neurocrine's website. Just refer me. No, I'm just kidding. But anyways, so in a nutshell, that is the system, and it has helped us make that iterative cycle much faster. So that iterative cycle that used to take weeks is now days. And even on projects where I've worked with different biologists, I've finished the whole project chemistry in 2 weeks, and that typically would have taken 2 years with multiple chemists. So the success is there. It's not just that you can make 9,600 compounds a day, it's that you can make the right compounds when you need them. So that's it.
Does anybody have any questions for Derek? And if you don't mind, just maybe repeat them back for everyone else.
Repeat.
When they ask you a question.
Oh, yeah. Okay.
I have a question. So you showed a lot of liquid handling, but in chemistry you have a lot of inorganic salts, bases, catalysts that you need to add that they do not dissolve in NMP. You need water. How do you add that and get rid of it?
How do we add water?
A lot of hygroscopic reagents or the inorganic salts.
So salts, we usually we freebase them before we turn them into solutions. So, or we, anything that's not soluble, we leave out of the system. Or we treat them as a slurry when we dispense them. So, sorry, she asked, how do we treat salts and solids, essentially, right? Hygroscopic compounds, again, any compound or any chemical that is sensitive to air and stuff like that, we don't put those in the system. We could have built a system to manage that, but those are 10%, 20% of the reactions, or 20% of the chemicals that we use, and we figured it's better to leave them out and do those by hand still. That way we don't have to build a really complex system. So.
Hey there. So do you have any plan, or do you require new reagents? Do you send 20 ml vials for the vendor?
So when we do buy new chemicals, we actually try to push as much of that through e-molecules. Sorry, Nady asked, do I send 20 ml vials to vendors to put their chemicals in? Ideally, that would be the situation. Ideally, I would like to do that, but most vendors don't offer custom solutions. So that's where we go to e-molecules, because e-molecules will actually order the chemicals for you and have the chemicals shipped to their site, and then they will reformat them into any vial that you want. So I actually do work with them to aliquot out solids and make our solutions for new chemicals.
And so what's keeping you from running the assay, the biological assays on?
So Nady asked, what is keeping me from running biological assays? So the only thing keeping me from running biological assays, because liquids are liquids, whether they're chemicals or biological reagents, is the biologists don't want me to.
Any other questions? Yes.
So this Verso unit is only for the chemical storage, or is also the biological samples you can also use there?
So the Verso is vial storage or plate storage. It's an SBS footprint style system. So it can store chemicals, it can store final compounds, it can store biologics. I believe they can make Versos cold to -20 degrees. Is that right? So you can pretty much store anything that's in a vial.
Quick blurb about our booth, by the way. Hopefully everybody's seen it. If you've been to the exhibition hall, we're right there in the front. 805 is our booth number. So we have a bunch of people on hand to answer those questions, go into more detail if you'd like. I have a question for you. So this was largely like your vision to bring automation to chemistry. Do you have like another new vision?
So, as I was saying that this is modular, we have actually purchased an extension for this. We're going to add 2 more modules because we're retiring our matrix store, which holds all of our final compounds. So we're going to compress what's in the matrix store, and we're going to store it into the Verso when we expand it. And then we're also hanging another Vantage off of it to do compound management from purification. So when our compound management, or when our purification group gives us dry samples, we're going to automate that process and make that workless driven. And we're also going to store our compound library in there in deep well 3-4 well plates. And then we'll make aliquots for acoustic dispensing when we need those. So. But it will all be done through the same style work list database.
So question, I'm going to talk about this, but why would you want to expand on that, being modular and all, but instead of having a similar system until you have redundancy in case one goes down, since you're already planning on doing the same thing on the other end?
So Nady asked, why wouldn't we just build a redundant system, right, to do the other process? Well, Neurocrine learned its lessons on spending lots of money, and we try to be as efficient as possible. And the system is pretty reliable. So if we had to worry about redundancy, I don't think we would probably have gone with Hamilton as a vendor. So the one major factor is this thing had to work. So. No, but it works. I've run 4 or 5 day audits on our Verso straight for 24 hours a day. It doesn't stop, so it works. And the largest chemical run I did was 80 hours. The only thing I learned is we didn't configure the system right because my trash overflowed inside of the Vantage too fast, and I had tips spilling out of the deck. So, there you go.